మీరు డిసేబుల్ చేసినట్టు వస్తుంది సార్.
రైట్ మేడం, ఇప్పుడు చూడండి.
మీరు ఇప్పుడే రికార్డింగ్ పెట్టుకోకండి సార్, అంతా
రైట్
అయిపోయింది.
ఓకే యూ కెన్. ఏ వెరీ good afternoon, my dear young friends. So today's our eminent resource person, Dr.P.Savita Madam Garu, Assistant Professor, Department of Botany, Head of the Department, GDC Luxetipt. Madam is going to explain about the gymnosperms. It is one of the important area of the plant sciences for all competitive exams, including CPGET-2023, CUSET, JL, DL. Even though SET, CSR, NET also, gymnosperms are the most important area for the fetching marks, and you will get good rank in the all competitive exams also. So we would like to invite today's our speaker, Dr. Savita Madam Garu. Please welcome and start your session, Madam. Please welcome.
Thank you, sir. Good afternoon, everybody. Good afternoon. So today we are going to learn about the life cycle of Gnetum. So here previously we have learned about the life cycle of the pinus. We have seen the formation of the cones in the pinus, and after these gymnosperms we'll be seeing the angiosperms, where these are the flowering plants. So this Gnetum will be having some more advanced characters in the gymnosperms, which resembles mostly the angiosperms or the dicot plants. So many characters are there in the gymnosperms which resemble the dicot plants. So listen carefully, students. If you have any doubts, you can ask me. So when you come to the classification here, division gymnosperms, class Gnetopsida, order Gnetales, family Gnetaceae, genus Gnetum. So the classification is very easy. Starting from the class, you will be seeing the Gnetum only, Gnetopsida, Gnetales, family Gnetaceae, and genus Gnetum. And when you come to the next distribution of the plant, distribution of these Gnetum plants, they are distributed in tropical rainforest in the equatorial region, African tropical forest, and Andaman and Nicobar Islands in India. They are distributed in tropical rainforest and northern equator and African tropical forest and Andaman Nicobar Islands in India. It is not in North America and European countries and also in Australia. It is not seen in North America, European countries, and Australia. So Gnetum plants, they are mostly distributed in tropical rainforests. Next, when you come to the morphological characters of the Gnetum, it is a sporophytic plant. The stage of the plant is diploid stage. So alternately, we will be seeing the gametophytic stage also, but the predominant stage is the sporophytic stage, which resembles the angiospermic plants, where three types of plants can be seen in the Gnetum. They are trees, shrubs, and climbers, woody climbers, which are called as the lianas. So here, in the previous slide, we were seeing about the plants here. This is the male, first one is the male plant here. First one is the male plant. This is the male plant, and I'm seeing about the pointer option here. Wait a minute. So here, this is the male plant of the Gnetum, and this is the female plant of the Gnetum. So when you come to the lianas, these are the woody climbers. Here in the diagram, you can see the woody climber, which is twining around the other host with the woody stem. So here three types of plants. Trees, where Neton is the example, where for the lianas, it is the Gnetum ula and africana. So most of the plants in Gnetum are lianas only. They are woody climbers only. Some may be bushes, some may be herbs, or some may be shrubs. So most of the plants in Gnetum are lianas only. Most of the species of the Gnetum are lianas, are woody climbers. They twine around the host with their woody stem. And some may be shrubs and some may be herbs. So when you come to the examples, Gnetum ula, Gnetum latifolium, Gnetum montanum, they are the lianas. Gnetum ula, Gnetum latifolium, and also africanum also. Gnetum ula, Gnetum latifolium, Gnetum montanum, and Gnetum africanum, they are the lianas. And Gnetum limon, Gnetum contractum are medium-sized trees. They appear as dicot trees only. Gnetum trinor, which is a parasitic plant on cinchona. This is very important character. So regarding your competitive examinations, so Gnetum trinor is parasitic plant on cinchona. This is about the morphology, what you say, the habitat of the plant. And when you come to the root system, it is taproot system with well-developed lateral roots and root hairs. It is taproot system with well-developed lateral roots and root hairs as in dicots. When you come to the stem, aerial, erect, woody, branched, and bear two types of branches. Stem, it is aerial, erect, woody, branched, and bear two types of branches. These two types of branches are dwarf shoots or branches of limited growth and long shoots or branches of unlimited growth. Dwarf shoots are branches of limited growth and long shoots are branches of unlimited growth. In lianas, internodes of the stem appear swollen in nature. Around the internode, you will be seeing swollen areas and on either side of the internodes in the lianas. When you come to the leaves, two types of leaves are present. They are scaly leaves on long shoots at nodes in opposite decussate manner. When you come to the leaves, two types of leaves will be present, scaly leaves and foliage leaves. These scaly leaves and foliage leaves both are arranged in a decussate manner, in opposite manner. Opposite decussately arranged leaves are present and the scaly leaves are present at the nodes. And in this, from the axils of the scaly leaves, you will be seeing that foliage leaves are formed and these foliage leaves are stipulate, petiolate, simple, with a thick leathery texture, oblong or elliptic. Elliptic means they may be either elliptic in shape or oblong, like a the banana leaf, or with entire margin, with a simple margin, without any extras like a serrate margin or etc. Entire smooth margin will be there and with reticulate venation. This is about the leaves here. And when you come to the anatomical characters and the two root anatomy, young root show three distinct regions, mostly resembling the dicot roots. First one is the epidermis. It is single layered without intercellular spaces. Here in the diagram, you can see the epidermis is single layered with a cuticle above it and without any intercellular spaces. Root hairs are present, and below this epidermis, you will be finding the cortical region, which is differentiated into two regions only. The below the epidermis, the region is filled with the parenchymatous polygonal cells. Parenchymatous polygonal cells, which are arranged very near to each other without any intercellular spaces here also. So this is the cortical region, and in another region in the cortical region is the endodermis, which has a single layer of parenchymatous cells without any intercellular spaces, which is parenchymatous in nature, which is single layered in structure and without any intercellular spaces. Endodermis, which helps in storing the starch materials, and this cortical region also helps in the storing the starch materials. And below the endodermis, you will be seeing the vascular cylinder. This so outer epidermis, which is single layered, which is covered by cuticle with the root hairs, and cortical region is divided into only two regions, that is outer one, which is present below the epidermis, which has the polygonal cells, which are arranged very near to each other, and endodermis, which is also a single layer structure with parenchymatous cells. And when you come to the next part, that is the vascular cylinder. Here the vascular bundles, they are a pericycle. Outer one is the pericycle, then vascular bundles, then medulla will be present. Here the pericycle, it is 2 to 3 layered, which helps in the formation of the lateral roots. Pericycle is 2 to 3 layered parenchymatous structure, which helps in the formation of the lateral roots. And when you come to the vascular bundles, you will be seeing that vascular bundles, they are only 2 in number. Only 2 vascular bundles. There are radial vascular bundles here because it is a root. Radial vascular bundles are seen, and they are, that is diarch condition, means only 2 vascular bundles will be seen, alternating with each other. Phloem and xylem are arranged on alternate radii, on different radii, alternating with each other. So they are exarch in nature, where the protoxylem is facing towards the epidermis. Here exarch and diarch condition of the vascular bundles is seen, which alternate with each other, and medulla will be very small and it is parenchymatous in nature. So this is about the vascular bundles. And when you come to the secondary growth, it is as usual as we see in the dicots and other plants, advanced plants. So during the secondary growth, we see that between these vascular bundles, radial vascular bundles, whatever parenchymatous tissue is present in between the xylem and phloem, from that a cambium will be arising, which cuts off xylem towards inside and phloem towards outside. So this forms the secondary growth in the stellar region, where in the cortical region a cork cambium forms from the cortical cells, and it cuts off cork towards outside and secondary cortex towards inside. So here in the secondary growth, a small mistake has been given in the PPT. So phloem inside and xylem outside, I have written that, but xylem towards inside and phloem towards outside. So this is the about the secondary growth, which is as usual as in angiospermic plants. Very clear characters in Gnetum. You can see very easy characters and clear characters. So when you come to the anatomy of the stem, then when in young stem of the Gnetum resembles dicots in structure, and it has 3 parts, that is epidermis, cortex, and the stele. Epidermis is single layered, which is thick parenchymatous without any intercellular spaces. And here you will be seeing that it is covered by a thick cuticle, and sunken stomata are present. Here in sunken stomata are present, single layered epidermis with a thick cuticle, no hairs are present. And below the epidermis, you will be finding the cortical region, which is divided into 3 regions: outer cortex, middle cortex, and inner cortex. Outer cortex, middle cortex, and inner cortex. Outer cortex, which has the parenchymatous cells with chlorenchyma, with chlorophyll, chloroplast, etc., which helps in photosynthetic process. Outer cortex, which is parenchymatous and chlorenchymatous, which helps in photosynthetic process. Middle cortex is many layered in structure, and it helps in the storage of the starch, etc., reserve food materials, etc. And in the inner cortex has the sclerenchymatous cells. Sclerenchymatous cells in the inner cortex, which are also called as stone cells, which are also called as spicular cells, which is also called as spicular cells. And mostly these spicular cells are thought to be the endodermis. No conspicuous endodermis is present in the stem of the Gnetum. We won't find any endodermal region separately here. So some of the scientists, they thought that these spicular cells are the endodermis of the Gnetum. And when you come to the vascular cylinder, here centrally located vascular cylinder is there. A pericycle is also not distinguished clearly, and 12 to 24 vascular bundles are arranged in a coral-like fashion, where the vascular bundles are conjoined, collateral, and open type, and endark in condition. Open type, collateral, conjoined, and endark condition. Open means here cambium will be present, which helps in the secondary growth. And this, when you come to the protoxylem part, it is present towards the medulla. Hence, this is called endark in condition. As usually we know that the roots will be having the xylem, exarch xylem, and whereas the stems will be having the end-arch xylem. So this in between the xylem and phloem, a cambium will be present, which helps with the secondary growth. And these vascular bundles are arranged in a whorl-like fashion. So this is about the primary growth of the plant, and well-developed medulla is present, where this medulla is made up of parenchymatous cells, which extends between the vascular bundles as medullary rays. So a well-developed medulla will be present, which is parenchymatous in nature and which extends in between the vascular bundles as the medullary rays. This is about the primary growth of the stem. And when you come to the secondary growth, in the secondary growth, some important characters you will be seeing in lianas. So when you come to the secondary growth, it takes as usual in dicot plants, as usual like dicot plants, by the formation of the intrafascicular cambium, by formation of interfascicular cambium between the intrafascicular cambium, joining the intrafascicular cambium, forming a cambial ring. We know that the cambium present in between the primary phloem and primary xylem is called as the intrafascicular cambium. During the secondary growth between the vascular bundles from the conjunctive tissue or parenchymatous tissue present in between the vascular bundles, a cambium will be formed, which is called as interfascicular cambium. And this interfascicular cambium which joins the intrafascicular cambium, forming a cambial ring, which cuts off xylem towards outside, inside, and phloem towards outside. So by this, the stellar secondary growth will be taking place. And when you come to the cortical secondary growth, some of the cells in the cortex, they act as the cork cambium, and they cut off cork towards outside and secondary cortex towards inside, like angiosperms only. And this here in xylem consists of xylem vessel, tracheids, vessels, and xylem parenchyma. Tracheids, vessels, and xylem parenchyma. And xylem will be having uni-apertures as in angiosperms. The ends of the xylem tubes, the ends of the xylem tubes, tracheids will be either it may be either if it is a vessel or a tracheid, the ends of this tracheids of the vessels will be having uni-apertures, only single aperture is present. And this is similar to the angiospermic character. And when you come to the phloem, it consists of the sieve tubes, phloem, parenchyma, and companion cells are absent here. So in roots, companion cells are present, but in stem, you will be finding that companion cells are absent in the stem. So phloem consists of sieve tubes, phloem, parenchyma, and companion cells are absent. So sometimes these sieve tubes may be associated with some parenchymatous cells, but they are not equal to companion cells because they are not formed from a single initial cell. In angiosperms, we see that the sieve tubes are associated with the companion cells, and these two are thought to be ontogenetically similar because they have arisen from a single mother cell. But in this Gnetum, you will be seeing that the phloem parenchyma, the sieve tubes, even though they are associated with the parenchymatous cells, these are not thought to be as companion cells because they are not formed from a single mother initial cell. So sieve tubes and phloem parenchyma are present in the secondary xylem of primary xylem. Secondary growth in the cortical region develops outside and develops a cork cambium, which cuts off cortex, secondary cortex inside and cork outside. And this is about the secondary growth in Gnetum pneumon, which appear like trees in structure. So, but when you come to the lianas, somewhat different secondary growth will be seen, where in this, when the secondary growth starts, it starts usually by the formation of the cambial ring between the vascular bundles, as usual, where it cuts off xylem towards inside and phloem towards outside. And in the cortical portion, cork cambium forms and cuts of cork towards outside and secondary cortex towards inside. This is the usual normal secondary growth in in the starting start time. But after some times, this function of the cambium stops, and from the secondary cortex, another cambial ring forms, which cuts off xylem towards inside and phloem towards outside, forming another secondary cambial ring in the cortical portion. This functions for some time, and again after some years, another cambial ring will be formed. Like this, we will be seeing at least 2 to 14 cambial rings, which are called as eccentric rings, are formed during the secondary growth in Ula and Africana. These are lianas. So a anomalous secondary growth, we can say it is a different type of secondary growth in this species. This is about the secondary growth, and these are all about the anatomical and morphological characters and distribution of the Gnetum. So this today I thought to stop here only. Reproductive characters we will be discussing tomorrow. If you want me to explain it in Telugu, or if you want me to explain it again, I'll explain it. Sir? Excuse me, sir.
Yes, madam. Regarding of this class, my dear friends, have you any doubts? Okay. Madam, it is clearly understood about the morphology of the details, the type study, Gnetum is clearly explained, madam. Remaining will tomorrow. Okay, madam? So reproduction of the Gnetum and the life cycle, the entire life cycle will going to explain tomorrow. Okay, madam. Thank you very much.
Good point, sir. If anyone wants me to explain it in Telugu also, I can explain it.
So, madam is also asking you, my dear friends, if those who are need Telugu also, madam is going to explain in both language, Telugu and English. If anybody is interested to listen in Telugu also, madam can explain in Telugu also. Okay? Right, madam. Tomorrow onwards, some of the participants in previous classes, they are already asked, madam, but they are today not joined because of some of the Telangana Dashabdi Utsavala celebrations are going on in different colleges. That's why they are unable to join. So definitely meet next class, madam. Thank you.
Okay, thank you, sir.
Right, madam. Right.